Hi everyone, welcome to your first Biology 105 video lecture. A couple of things I want to remind you about. First of all, if you are viewing this video lecture without a pen or a pencil in your hand to take notes, then you are doing it wrong. One of the benefits of having these video lectures is that you can pace them for yourself. So if you need to pause while you take some notes, then you should do that. Whatever note-taking system works best for you, but I promise the action of taking notes while I'm talking to you is going to help you remember this information so much better. Second thing I want to point out for you is that you do have a blank version of this PowerPoint available to you on Blackboard if you want to use that for note-taking purposes. Also, don't forget, this corresponds to Chapter 1. I try to kind of follow the outline of information in Chapter 1 very closely, so you should definitely refer to your text if you want more information, but sometimes I'll include little tidbits of information that I know. And so you are responsible for both the things I say and the things in the chapter. And then the last thing I want to point out is that sometimes I'll have little notes for you here. Let me see. Get this. Oops. Okay, sometimes I'll have notes for you here in this note bar, okay? If you're looking at the PowerPoint and you're not sure how to access notes, you've got a little notes tab here that you can click on, and then you can scroll up or down to see anything that I might have added. I don't have notes on every slide. Sometimes I have them for you though, okay? All right, I think that we are now ready to go ahead and move forward with our lecture. So this is going to cover Chapter 1, which is really just an introduction to biology as a discipline. So essentially what we're going to do in this chapter is talk about biology as the study of life. And in particular, we're going to focus on two big picture concepts. First, what constitutes a living thing? And second, how is it that we go about studying biology, okay? So in thinking about what constitutes a living thing, there are 5 characteristics that we want things to have. I'm going to put, that's an abbreviation for characteristic. 5 characteristics that we want things to have in order to decide that they're alive. Now, while I'm going through this bit of the lecture, I want you guys to be thinking about viruses, okay? And we can talk more about viruses at our in-person lectures, but don't know how much you know about viruses. It might be worth Googling them. As we're going through these 5 characteristics of life, I want you to think about whether or not viruses satisfy these characteristics and whether or not you think they're alive or not, and are they included in the discipline of biology. All right, and then the study of biology is simply going to be us reviewing and going back over how science is done, which is essentially going to be a discussion of the scientific method. And my hope is that you have some familiarity with that based on your previous studies. All right, so let's first focus in on these 5 characteristics. So what is alive? What do we require as biologists to determine that something is alive? Well, first and foremost, we do expect it to have or be made up of cells, 1 or more. It can be unicellular, it can be multicellular, but we do expect it to have a membrane-bound membrane-bound cell. And we believe that this is the basic unit of life, okay? All of our evidence to date supports this. All right, next. We also feel that for something to be alive, it must replicate or reproduce. I'm going to put replicate, but reproduce would work just as well. So it's got to be able to make copies of itself, right? It also must consume energy in order to sustain itself, right? To be alive, things require energy, so we're going to talk about that. What else? Well, one of the other requirements of things that are alive is that they must store and transmit information. Now, what do I mean by that? Well, we have to have a set of instructions that tell organisms how to function, right? So you've got to store information that tells your body how to function, and then you also have to use that information and access that information in order to actually carry out your body's biological processes, right? So we're going to talk a little bit about information. Hopefully you're guessing that I'm talking here about genes, right? Genetic or hereditary information. And then the last thing that we believe life requires is that it evolves. It changes over time, okay? So this lecture is going to take a closer look at each of these 5 attributes of life. Let's begin by talking about cells. So we didn't always understand that all living organisms are made up of cells. It wasn't until sometime in the 1600s, around 1665, that there was a scientist named Robert Hooke, all right? And so what's his deal? Well, what Robert was able to do, and it's actually H-O-O-K-E. All right, so what was Robert able to do? Well, he devised, sorry about the return here. He devised a microscope that was actually able to magnify things to 30 times their original size. That probably doesn't sound like very much, but when you're used to seeing things at their actual size, you would be surprised at the level of detail that you can get at just using a 30 times magnification. And what Robert began looking at in particular, he was looking at some very thin slices of cork. You know, like the stuff that's in wine bottles, right? Basically what that is, it's a very soft wood fiber, okay? So he was able to take like a razor blade and slice this cork into really, really thin sections. And that's what you're looking at in this picture here, okay? What he noticed immediately was that this cork tissue seemed to look like a grid, and hopefully you can make out that you've got, like, let's just hone in here, you've got a series of block-looking structures, don't you, okay? And when he was looking at these, he thought that they reminded him of rooms, and in particular, he thought they reminded him of rooms in a monastery, and the rooms in a monastery are called cells. So that's what he called these things here that he was seeing, okay? He called these cells. Now, the great irony is he wasn't actually looking at cells. The cork tissue that he was looking at was dead, right? Wood that you have in your backyard that's fallen out of the tree and you would maybe burn for a fire is usually dead. What does that mean? Well, basically it means that all of the guts of the cell, things like the nucleus or the Golgi apparatus, or any of the stuff that you learned about in 5th grade science, all those cellular parts were dead and gone. And the only thing that remained was the outer structure here called the cell wall. And hopefully this is ringing a bell for you from earlier biology classes. The cell wall is simply a rigid outer structure. In plants, it's made up of something called cellulose, which is a cellulose, which is actually a structural carbohydrate. Oh, misspelled, there's an L in there. Sorry. All right, cellulose. So that's the stuff that makes up wood, and that's all that was left was this hard outer structure, this cell wall structure. So it's kind of ironic that we get the name for cells from what he was seeing, but he wasn't actually seeing cells. Now, what's interesting is we now have one observation of cells by one guy, right? That hardly constitutes a statement like all living things are made of cells. One guy's seen them in one spot. But over time, we began to see more and more things that appeared to be made up of cells. So enter this guy here. There was a guy in 1665. So this guy was, oops, this is actually 1673. I didn't change the date. Lies. Let me fix that for you. 1673. I'll try to remember to fix that in your PowerPoint. Anyway, a few years later than Robert Hooke. This guy's name, he was kind of an interesting cat. His name was Anthony, and he was Dutch. So this is the spelling. Anthony van Leeuwenhoek, and I'm going to put an L. The spelling is actually going to be in your notes, okay? It is a monster to spell. I'm not Dutch. So Anthony van Leeuwenhoek is how you say that. And he was an interesting guy. He was a tailor, and cloth was like a big deal industry in the Netherlands in this time period. So he was a rich, rich dude. And one of the things that determined the value of cloth in that time was the thread count, how many threads you would see like in a little piece of cloth, right? Some of you may have experienced this when you're buying sheets or something like that. Something with a high thread count is very supple and soft. So the higher the thread count, the better the quality of fabric. So when he was making his microscopes, he wasn't so much interested in looking at biological matter. He was looking at cloth samples. But he was actually able to devise a microscope that was 10 times stronger than Robert Hooke's. So he was at like 300 times resolution. And this is an example of a Van Leeuwenhoek microscope right down here. The way it would work, so I don't know if you can make out that right there is a tiny little lens, right? And what he would do is he would put a little drop of pond water, okay, here. And then there would be a candle over here somewhere. So I'm gonna draw a candle, shoop, shoop, candle. Here's a wick, and there's a flame. I'm such a good artist. Okay, candle here is a light source. So your eyeball would be on this side, right? And you'd be looking on this side through here, and you would be able to see whatever was inside this little drop of water, and it would be magnified. So he saw lots of things. One, for example, he saw a lot of little tiny organisms that he called animalcules, and animalcules, okay. And in addition to that, he saw a lot of other cells. So we're beginning to accumulate more and more evidence for this idea that all living things are made up of cells, okay? You can't just say that based off of one observation, but as we begin to have more and more observations, now a theory is building. And over time, more and more people devised other microscopes, and we start to have this idea that cells are the thing. They are all the rage. Incidentally, these microscopes, you can actually go to museums in Europe and see these, and people today can't replicate the magnification that this dude was able to get with this very simple machine. So it's kind of fascinating. So over time, we accumulate more and more evidence, and the scientific community begins to realize that all living things are made of cells. But that's where things kind of stop. After about 150 to 200 years between Hooke and Leeuwenhoek, there was very little progress made in cell biology. People saw them, they're like, hey, they're there, whatever, what's the big deal? And they didn't really think a whole lot more about cell biology, and this is likely due to the very widespread belief in something called spontaneous. I'm going to try to spell while I talk. Generation. And I am just going to abbreviate that as spontaneous gen. All right, so what's spontaneous generation? You've probably heard of this. Now, remember that in this period of time, we're talking about the 1600s, we didn't have a public education system like we do today. Most folks were not educated people, and certainly the kind of community that would be talking about cells, the scientific community, was restricted to a very few individuals. What most people believed was what they saw with their own eyes. And what they saw with their own eyes was things arising out of nothing. In other words, they were spontaneously, which means all of a sudden, generating, okay? So they were just arising out of nothing. And so some examples are maggots from rotten meat, right? If you've ever had the experience of having maggots on something, they feel like they come out of nowhere. They're disgusting, right? And they feel like they come out of nowhere. You almost never see the little eggs, right? And if you didn't know that flies laid these tiny eggs that are virtually invisible to the naked eye, you might believe that those maggots came from the rotting meat. Another thing that people thought was that mice came from hay or from wheat, right? So they wouldn't necessarily see the baby mice because they'd be hiding, but sure enough, if they went out into their grain storage or in their hay storage, they would see all of these little mice, right? So they thought, and this also jived with their religious beliefs, right? So we're talking about, particularly in Western civilization, at this point, Christianity had taken hold. You have this belief in this creator God who just created humanity and all of the earth out of nothing, right? So this really jived with what people saw and what people believed religiously. So there was this gap for about 200 years where there wasn't a great deal of progress made in understanding cells. But what really changed the game was in the 1860s, and I want to say this was 1862, there was a paper published by someone you may have heard of. His name was Louis Pasteur. If you've ever had pasteurized milk, you can thank this guy. So he is responsible for a lot of stuff. If you are a health pre-professional, like a pre-nursing major or pre-med, something like that, you'll learn more about Louis Pasteur in your microbiology classes. He's considered one of the fathers of microbiology. He was an early immunologist. He developed vaccinations against anthrax and cholera. But one of the things that he did was he also performed a very famous experiment that is known for debunking, or discrediting is another way of saying that. So he debunked spontaneous generation. Okay, hopefully you know what I mean. He debunked spontaneous generation. So what did he do? He devised this very, very elegant experiment. So the first thing he did was he took some broth, like just basically like chicken broth, right? This is actually really good for growing bacteria. Any broths, like if you ever take microbiology, you will use a series of broths to culture your bacteria. So be very careful when you're using broth at home. You want to definitely store it appropriately. So he took some nutrient broth and he put it in a glass flask and he boiled it. That killed any cells that were present in this broth, right? And then he let this broth sit over time, and lo and behold, cells reappeared, right? Cells reappeared. So this actually would seem to support spontaneous generation. You had cells, you got rid of them, you let it sit for a while, and boom, suddenly they reappear. But based off of the work of some previous scientists, Pasteur knew that the likelihood was that whatever cells were in here were the result of air transport. So there must have been some bacteria in the air here that, because this flask was open to the air, it could get down in here and cause these to grow. So to test his hypothesis that this wasn't spontaneous generation, that instead there must be cells from the air, he took and he heated up the glass in this region here. He heated it up and he stretched it out and made this area here. This is called a swan-necked flask, okay? And the beauty of a swan-necked flask is, you can imagine, if you've got any little bits and particles that are in the air, they might get in here. They're gonna have a tough time traveling up this swan neck and getting in here, aren't they? So now he repeated this experiment. He boiled this broth, got rid of whatever cells were in there. Now this is a sterile broth, and he let it sit and he let it sit, and lo and behold, the broth remained sterile because, as he predicted, any of the stuff floating in the air got caught in this swan neck flask. So make sure that you understand how this experiment works to debunk spontaneous generation, right? Hopefully you can see that here, it looks like spontaneous generation might be happening unless you understand that these cells didn't come out of nowhere. They came out of the air, and how he tested that hypothesis here. This is a very elegant experiment. This experiment was done in 1862, okay? Or actually, I think the paper where he reported. I don't know if this is exactly when he did it, but I think the paper came out in 1862. So we're talking like 200 years since Robert Hooke first saw cells. So this sort of led to the development of what we today call the cell theory. And the cell theory was actually proposed in 1858 by a guy who's in your notes down here, Rudolf Virchow, okay? But once Louis Pasteur's paper came out, people really started buying this idea of the cell theory, okay? The cell theory has basically got three parts. You need to know the 3 parts of the cell theory, guys, okay? So first and foremost, all organisms are composed of 1 or more cells, okay? So look here, all things are made up of 1 or more cells. We have a plant, one that's more like multiple cells, right? If we had an amoeba, that would be a unicellular beastie, right? But that still qualifies, that's 1 cell. We also agree that the cell is the basic unit of life, okay? It is the smallest unit of life. So here we have a unicellular amoeba. It's alive, it's got 1 cell. Here we have a flower, it's got multiple cells, but all of its chemical processes are actually happening inside these cells. So it's the basic unit of life. And then last, but certainly not least, is that all cells come from pre-existing cells. And this is a direct result of getting rid of spontaneous generation. Let's go back one. These cells here didn't come out of nowhere. They came from pre-existing cells, right? So this is no spontaneous generation. No spon gen, right? Cells have to come from somewhere. They're not just poof, magically placed somewhere, right? So there's actually some implications to this right here. This third tenet of the cell theory has some implications. So it means that cells have to replicate for life to exist. And it also means that the cell theory predicts that we are all connected by a common lineage. What do I mean by that? Well, if all cells come from other cells, then that means there must have been a cell prototype way back in early evolutionary history that gave rise to all of the life that we see today, right? Because all cells must come from other cells. Now, that probably begs a question for you. Okay, that sounds great. Where did this first cell come from? Where did the first cell come from? And we're going to spend some time talking about that, okay, where this first cell might have come from, how it might have arisen. There's some different theories on how that might have occurred. All that happened, though, it just had to happen one time. And once it did happen, then this could replicate and we could end up with the diversity of life that we see today. And we have our earliest evidence of cells from about 3.8 billion years ago. Now I want you guys to go and look up, how old do we think the Earth is? Okay, and we'll talk about this in class when we go over this lecture. How old do scientists estimate the Earth is? And you'll get some idea that life has been present on Earth for the majority of what we believe to be its history, okay? I say believe, but we have evidence of its history as being, okay? So that is cells. Now, these cells, in order for these cells to exist, they're going to require energy, right? Cells, basically, as the basic unit of life, what they're doing is they're undergoing all of these chemical reactions that allow the organism to live and have metabolism and do the things that it does, right? So all of the chemical reactions that take place inside a cell require energy. And you've probably heard of the type of energy that cells use. The currency of energy, okay, like the money, the energy money of the cell, is ATP, which stands for, in case you didn't know, adenosine triphosphate. We're going to talk a whole lot more about this later. But that is what this picture here is, okay? So this is the adenosine part, right? And then you've got, these are called phosphate groups. And look how many we have. 1, 2, 3. Adenosine triphosphate. And so this is the chemical energy that the cell will use in order to accomplish its cellular chemical reactions. And that's because, for reasons we will discuss later, there is a lot of energy stored in these phosphate bonds. So that's one type of energy that cells need. A second type of energy that cells need is they need some building blocks, some Legos, if you will, in order to make other cells. So things like DNA, RNA, and proteins, okay? So cells require energy. Now, in thinking about how cells get that energy, different types of cells have different strategies for getting this energy. So plants, some bacteria, and a group of prokaryotes called archaea, which if you haven't heard of, we'll talk about some more later. They produce sugar using energy from chemicals or from sunlight. You've probably heard of photosynthesis, right? And I'm just going to abbreviate that, photosyn, because it takes me a while to use my tablet to write. So you've probably heard of photosynthesis, and you know that that's the business of taking light energy and making it into chemical energy of ATP, right? Let's draw light. And then we use photosynthesis to convert that into ATP, right? In this way, we're able to harness this light energy, make it into chemical energy. Another strategy that some bacteria and some archaea use is they can actually take chemicals that have high energy bonds and break those bonds and use a process called chemosynthesis. This happens in the depths of the ocean where there's no light. Okay, so chemosynthesis, and then chemosynthesis will take that chemical energy that's stored in chemical bonds and then convert it to a different set of chemical bonds in ATP. So this strategy for acquiring energy is used by what we call autotrophic cells or autotrophs. And hopefully this is a term that you've heard before. And if you broke this term down, the suffix troph means feeding, and the prefix auto means self. So autotrophs are self-feeders. They're able to generate their own ATP by converting chemical or sunlight energy into ATP. Now, you guys know that we can't do that, right? Animals, fungi, some bacteria, and some archaea, we're unable to do this. So what we do, our strategy is to consume the critters that do. We consume these guys or other critters. So we have to get our energy by consuming other organisms. So we are called heterotrophs. Okay, so if troph means feeding and hetero means different or other, then we are other feeders, okay? All right, moving on. Let's talk about the next characteristic of life. So far we've talked about 3, right? Let's go back. Characteristic 1 is that all living things are composed of 1 or more cells. Characteristic 2 is that those cells have to be able to replicate, right? Characteristic 3 is that they require energy. So let's just go back through. We've got characteristic 1, which was our, let's see here, where's our cell theory? Yeah, characteristic 1 is our cell theory, right? Characteristic 2, we talked about how cells replicate, and this is the basically results in all of life being descended from a common lineage. And then characteristic 3 is that all cells require energy. So let's move on to characteristic 4. Living things, whether they're unicellular or multicellular, must store and be able to use information, okay? And we'll talk more about this later, but basically we know that this information is stored as genes, Okay, this is based on our chromosomal theory of inheritance. This is stored as genes, and genes are located on chromosomes. So let's take a look over here. Hopefully this is a familiar type picture to you. You can make out we have a series of cells here. I think that I have for you, I don't think these are plant cells, but I'll have to go back and check. They're kind of square though. Not sure. But what I can tell you for sure is that these are some cells that are undergoing cell division, right? So here we have a cell that's not dividing. Here we have a cell that's about to divide. The chromosomes are condensing, and here the chromosomes are moving apart, right? We've got a metaphase set here. This looks a little bit like we're getting toward telophase, but point being, can you see all these little stringy bits here? Those are your chromosomes. Those chromosomes are where genes are located, right? You guys have all heard of genes. So these genes are what equal the information that we're talking about. Think of these as the instruction manual that's going to tell cells or organisms how to function and how to replicate, right? So let's go into a little bit more detail. I get really excited about this stuff because I do a lot of genetics research. So we're going to talk later in the course in more detail about genes and DNA. But basically, chromosomes, where the genes live, chromosomes are made up of deoxyribonucleic acid, which you guys know as DNA, right? Okay. DNA is a molecule that's shaped like a spiral staircase. Can you see that here? Here we go. Right? It looks like a spiral staircase. And we call this shape a double helix, right? So we have two backbones and that are twisted up against one another, right? And if we use this analogy of a spiral staircase, the stairs or the rungs here are made up of 4 building block molecules. And you guys have probably remembered this. You have A, T, C, and G. And that's adenine, thymine, cytosine, and guanine, okay? And you probably remember, so remember, this is what makes up a chromosome. Chromosomes are made up of DNA, right? So I'm just gonna make a little arrow here. That's what that is an arrow. Shoot, I'm really struggling here. Okay, that's an arrow. All right, so you probably also remember from your general biology studies in high school that when we're looking at a molecule of DNA, we've untwisted it to simplify it here, right? So we've got our 2 backbones, and ordinarily they'd be twisted into a spiral staircase or double helix shape, but we've untwisted them here. And what you can see is that the rungs are actually made up of pairs of molecules. We call these molecules bases. And these pairs pair up according to some very specific base pairing rules. So A pairs with T and C pairs with G. And so this actually is how it's really elegant. It sort of gives us an immediate understanding of how information is stored, because you can copy this information really easily. If you pull these 2 pieces apart, right, you immediately have a template to make a new copy. So let's look here. Because we know that A always pairs with T, here's our T, C always pairs with G, there's our G, et cetera, et cetera, et cetera. Now, can you see that this here restores this here? So because of the base pairing rules of DNA, we are able to store and copy. Let me put that here, store and copy information. Okay, this pairing allows us to store and copy information. And it also preserves the information that's encoded in DNA. All right. Now this leads me to, let's see here. Okay, one more thing I want to mention about the DNA. In thinking of DNA, remember how I said that chromosomes are made of DNA and segments of DNA equals genes. Let's write that out. Chromosomes equal DNA, right? That's what's made up. And if we take segments of this DNA, that equals a gene, okay? So if we wanted to diagram this out in a very simplistic fashion, and you'll learn that this is not exactly how things look, but if we want to diagram this out, then this whole thing would be a chromosome, right? And this would be gene 1, gene 2, gene 3, and gene 4, right? So the sequence of DNA here, this segment is one gene, this segment's another gene, et cetera, et cetera. And the way to think of genes is to think of the sequence of the A's and T's and G's and C's like letters in a word that tell the gene what it is, right? So it's the sequence of bases that are important here, okay? The A's and T's and C's and G's are what's important, and their unique sequence is like the letters that spell a unique word that equal a gene. And this is actually just a segment for a particular human gene called the human RAS gene, okay? Just to give you an idea. Now, so now we know that information is stored as DNA, and DNA is broken up into segments that are genes, and it's these genes that are gonna tell us how to do functions in our body. But that might not make sense to you. Like, how does this information here, how does this tell your body to like make blue eyes or to get your hemoglobin in your blood to carry oxygen? Like, how does this equal function? How is this an instruction manual? Well, that gets us to something called the central dogma of biology. Know this, love this, live this, preach this, okay? This will definitely be something I ask you about later. So how does DNA equal information to make organisms? Well, here is how. DNA gets copied into RNA. RNA gets made into proteins, and then proteins do the chemical work that lead to traits. Let's look over here. DNA is right here. DNA gets copied into RNA, okay? This is going to act as a messenger molecule. RNA is going to get made into proteins. Now, proteins are things like enzymes. And you might remember from previous classes that enzymes are proteins that carry out chemical reactions. So enzymes are able to do things like make red skin pigment in this newt. See how this newt is red? So it's these proteins that determine physical traits, okay? So for example, the reason that you might have brown or blue eyes is because you have a gene that codes for a protein or an enzyme that makes a brown or a blue pigment, okay? So DNA stores information for cells, but it by itself isn't enough. It has to be converted into molecules that can actually do the work of cells, all right? Make sure you understand this concept of the central dogma of biology. All right, so one of we've now gone through our 4 characteristics. We've got to talk about our last one, which is evolution, and it's directly tied to this idea of genes. So a mutation is equal to a change in the DNA, and it's mutations that lead to evolution. And I'm going to show you how that works. Okay. So remember that this is our central dogma. DNA gets copied into RNA, which then gets converted into proteins, and these proteins are changed into traits. We actually say that this is transcribed into RNA. We're going to talk about this later. DNA is transcribed into RNA, and RNA is translated into protein. All right, so this is our central dogma that we just talked about. But what if we made a change here? If we had a mutation, a change in the DNA here, right? Then wouldn't that ultimately cause a change here. And if we have a change here, so this is a change. We have a change in the DNA. We'll end up with a change in our protein. And that's going to cause traits to change, right? And this is how evolution happens. Let's look at a very specific example. Okay, so this is human chromosome 11. Not really. It's just an illustration of human chromosome 11. But there is a gene on human chromosome 11 called the beta globin gene. And in a healthy person, the gene sequence is quite long. The sequence of the A's and C's and T's and G's is quite long. But there's a portion of this sequence here that in a healthy person reads GT, GC, T, G, C, C, la, 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 la. You get me, right? But through mutation, through some accidental copying, there has been a mutation that was passed on into some folks, okay, where this T was accidentally copied in a way that converted it to a C. If a person carries this mutation, then they have something called beta thalassemia, which you probably have heard of as sickle cell disease, okay? So a normal person has this right here, and a sickle cell person's got this right here. Now, why is that important? Well, a normal person has functioning red blood cells that carry oxygen quite well and can flow through your blood vessels quite well. A person with sickle cell disease, these are misshapen blood cells and they're not going to flow through your capillaries as well. Therefore, oxygen is not going to be distributed or picked up by these cells quite as well, okay? So you can imagine that this now becomes a trait that evolution can act upon. So let's talk about what that means. What does it mean if evolution is acting upon a trait? Well, I am going to change gears now. We're going to talk about a hypothetical trait. So evolution is just a change in a trait in a population over time, okay? And this change comes about because of genetic variation. So let's look at this example I've given you here. In my hypothetical example, I have a population of little mice. And these mice come in two flavors. There must have been a gene mutation, right? So there was a gene mutation. Maybe these guys started out tan, or I don't know, they started out gray or tan, but there was a mutation and now we have some mice that are tan and some mice that are gray, okay? They live on this gray rock. This is a predator, okay? Well, because we have a mutation whereby we have two different colors, can you see now that we have a situation where this predator is able to see some of these critters better than others? So what happens is he eats the tan ones because he can see them, right? Now, once he eats these tan ones, are they going to be able to pass their genes on to the next generation of mice? Nope. Once you're dead, you don't make any more babies. You can't pass that tan gene on. So now what that means is the mice that are left to reproduce are 3 gray mice and 2 tan mice. So in our next generation, we're going to have a higher proportion of gray mice compared to tan mice. And then if he comes along and he eats these guys, and we do it again, over time, you may end up in a situation where you have no tan mice and all gray. Oops. Right? This is how evolution works. So we started out, we had a gene mutation where we have 2 different colors of mice. But it turns out that the mutation made these guys easier for the predator to see. And so over time, our population evolved to be all gray, okay? So that's how genes are related to evolution. So there's a couple of vocabulary terms I want you to know. First of all, this idea of a population changing over time, it happens through a process called natural selection. So let's look at your notes. This is a great example of when you want to look at your notes. Natural selection, as traits that lead to increased reproduction, become more common in a population over time. So let's see here. What trait is leading to reproduction? Well, if you're gray, you stand a greater chance of surviving the hawk and reproducing over time. So natural selection says that you have a greater probability of passing your genes on, don't you? So evolution happens through natural selection. Okay, now what determines the direction of natural selection? Well, that is determined by adaptation and fitness. So the reason I have them on here is I say that adaptation and fitness leads to natural selection. An adaptation is simply a trait that increases the ability of an organism to reproduce, okay? So an adaptation is a trait, can't write today, a trait that increases reproduction, right? Right? So you can see that the trait that increases your ability to reproduce here is being gray. So gray coat color equals our adaptation. Adaptation. Okay. So what's fitness? Fitness is just your ability to reproduce, okay? And I'm just gonna put repro. Sorry. Oof. Sometimes it's hard to write with this stylus. All right, ability to reproduce. So clearly the gray mice have greater fitness than the tan mice do, okay? So I've got these definitions for you down here. Make sure that you understand those. I usually ask questions about those. All right, so at this point, we've covered all 5 characteristics of life, right? Number 1, all organisms are made of cells, right? Number 2, all organisms need to be able to reproduce. Number 3, all organisms require energy. 4, they must be able to store and transmit information. And 5, they must be able to evolve over time, okay? And you need to know some of the details that we talked about associated with each of those traits. The last thing I want to mention about trait number 5 is that evolution has led to the diversity of life. And I mentioned that a little bit earlier when I talked about the fact that we had one prototypical cell that divided into other cells. And through the process of evolution, we have come up over 3.8 billion years with a great diversity of life, okay? And that life falls into 3 domains, which we're going to talk about more, but make sure you know what these are: bacteria, archaea, and eukarya. And I want you to be able to give me examples of organisms from each kingdom, or sorry, from each domain. So go and look that up. What kind? Give me an example of a bacterium. Give me an example of an archaea. Give me an example of a eukaryote. Okay? All right, the very last thing I want to go over, and I'm not going to spend a ton of time on this. This will be the subject of one of your early labs if you're taking Biology 115 lab. But if you're not, I still wanted to go over this. I wanted to talk about biology as a science and the process by which we do science, okay? I feel like this is a very timely topic because a lot of the process of science is on display as we see in real time people going through and learning about the biology of SARS-CoV-2, which is the virus that causes COVID-19. And you know, there's been a lot of criticism over, well, at first people were saying this and now they're saying that, and to that I say that's right. In science, as we accumulate more evidence, we change our understanding. That's how science works. So it's sort of like if you have a weather forecast You may not know exactly when it's going to rain, but as you get more information from radar and from surrounding areas, your forecast becomes more and more precise. And that's exactly how science works as well. Now, early in the study of biology, and even today, we still do a lot of what I call observational science. And what do I mean by that? So in observational science, you're going out and you're really just documenting what you see out there. So every time someone discovers like a new species, like I have a lot of friends who do work, my background is in marine biology. So there's a lot of friends I know that maybe go and they do sample from the ocean floor or whatever, and they go through and they sort all the different critters that they find in their ocean floor sample. And sometimes there's things that have never been seen before in there, and they describe those and they publish that information. And that's very much science, right? It's just that it's observational science. It's not experimental science. A friend of mine just finished her degree where she went through and she looked at all of the parasitic worms in several species of fish, and many of them were types of worms that had never been seen before. It's a great example of observational science. It's important information, but she didn't really perform an experiment. She just went out and documented what she saw. But much of the science that we do today is what we call experimental science, in which we formulate hypotheses and we use these hypotheses for finding evidence. So let's take a look at the scientific method here that I've got for you in this little picture. So the first step in the scientific method is that you make an observation, right? And then I would say that there is a step in between here where you develop a question based on your observation, right? I'm going to put develop a question. So here the baby is making an observation. Here is something, okay? Maybe the question is, I think it's good to eat, or let me see, that's a statement. Is this something good to eat, okay? So you form a hypothesis, an educated guess, right? And when we're talking about hypotheses, we are talking about, first of all, these are educated guesses. These are not just things out of the blue. So if you formulate a hypothesis, you hopefully have studied something and are making an educated guess about what's going to happen, not just any random guess, okay? And the really important thing about hypotheses is that they have to be testable. It has to be something that you can devise an experiment and test your educated guess, okay? So here, the baby's performing the experiment, which is eating the thing, analyze the data, it made him sick, report your findings, oh my goodness, and invite others to reproduce the results. Okay, so that's a really silly and oversimplified example, but let's take a look at what I think is an excellent example of the scientific method, and this is also documented in your text. So, in this example, there was a group of researchers that were studying desert ants from the Saharan desert. And what they found was that these ants had a really interesting foraging behavior. So they would leave their nest, and then to try and find food, they kind of meander around in this haphazard way, right? So wander, 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 food. Yay, okay. Once they found food, they didn't retrace their steps to get home. Instead, they were able to make a straight line back to their nest. So that's a really interesting observation, right? That may not be what you would expect. Like if they're out and they're just doo-ba-doo-ba-doo-ba-doo, wandering around, find food. Personally, I would have guessed that they would just kind of like retrace their steps to get home, but no, they take the shortest, most direct path home. So that's our observation. Your question might be, how do they navigate? Okay, that's a good question. How do they do that? How do they manage to find their way home? Okay, so, or how do they find their way back? So these researchers that were looking at these ants, they devised a hypothesis, and they said, you know what? We think it might be that they use stride length to measure the distance to their nest. Okay, so can they test this hypothesis of stride length? Well, it looks like they did, and I don't, this is sort of an interesting way to test this hypothesis. So what they did was they took these ants, and in order to test whether stride length was how they were finding their way home, like they counted the number of strides that they took and then kind of came home that way. What they did was they changed the stride length of the ants by changing the length of the ants' legs. Some of these ants, they cut off their legs to make little stumpy short legs, right? That's going to change their stride length. Their stride length is going to be shorter. For some of the ants, they actually built little stilts that they attached to the legs. So now their legs are longer than normal. And so their stride is going to be longer. And then they had a set of ants that they did nothing to. So they had just an untreated stride length, a normal stride length. So what did they do? All right, so far we have made an observation, right? And then in between we developed a question. And the next thing we did was we developed a hypothesis. And I'm going to say that this actually starts the beginning of developing an experiment to test the hypothesis, because I bet you can guess where this is going. So they're developing an experiment, right? What they're doing is they changed the leg length, right? And now they're going to let them forage. Now they're going to let them forage and find their way home and see if this altered leg length made any difference in whether they were able to get home. Okay, so they did this experiment. They let them forage. They went out. So these ants went out. Let me go back. So they went out and they found their food. And then what they did was they let them try to make their way home. This is their data. So now we're at analyze the data. So this is kind of the fourth step of the scientific method. Here we were at 1, 2, 3, 4. We've done the experiment. Now we're going to analyze the data. All right. So what's the data look like? Okay. So it looks like home is about 10 meters away. So they put some food out, and home is 10 meters from the food item. The stumpy critters, they kind of fell short of getting home, okay? They only made it about 5 meters away, and they never did make it home at 10 meters. The stilty critters overshot where home was. They went farther than where their home nest was. They went 15 meters, right? Didn't make it home. But the ones that they left alone made it home at 10 meters, okay? So we've analyzed our data. Now, that means that we can say something about our hypothesis. We can say that our hypothesis was supported, our hypothesis was refuted, or sorry, our hypothesis was supported, or our hypothesis was refuted. Either we think our data tells us that our hypothesis is correct, or our data tell us that our hypothesis is incorrect. We do not use the words prove. We never say that a hypothesis is proven, okay? So one of the questions I'll ask you the next time we meet is, does this data support or refute the hypothesis of the investigators? What is this data telling us? Okay, what do you think is going on? And the other thing I want you to tell me is, why do you think we say that hypotheses can't be proven? I've got that in the notes here. So answer those questions for yourself, and we'll talk about those a little bit the next time we get together, which I guess will be on Friday for everyone, okay? Now, one of the things that I want to point out, and this is really a question of language. Sometimes you'll hear people say things like, well, my theory is blah, 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 or that's just a theory. So in our common usage, sometimes the word theory is thought to describe something that has some uncertainty around it. But in science, that is not the case. When we call something a theory, what we are saying is that it's a hypothesis that has been tested over and over and over again. It has been supported and supported and supported and supported to the point that we're virtually positive it's true. But remember, we never say we can prove hypotheses, and I want you to think about why that is. So examples of things that we call theories in science would be gravity, the theory of gravity, right? We feel pretty good about that one. And evolution. Okay, when we say that evolution is a theory, we are saying that it is accepted as a virtual truth in science. It has so much support that it is considered to be true. In science, the word theory means something that is basically a scientific truth, okay? So it's a little different than our common language use of the term theory. All right, that brings me to the end of our very first lecture. So make sure that you've taken some notes. I've asked you some questions. Hopefully those will guide some of your study, and I hope to see everyone or as many folks that can come to class on Friday and answer these questions for me. All right, talk to you soon. Bye-bye.